Hello, everybody. Thank you for joining Certara today as we present a webinar on achieving virtual bioequivalence with PBPK in lieu of clinical studies for AAPS 2021. I am joined today by my colleague Nikunj Patel, who is a senior consultant and scientific advisor at Certara, and I am an SVP with the SimSip division of Certara. So thank you again. Just a little bit about Certara. I'm sure many of you are familiar with our company and many of the software products such as SimSip, additionally for Phoenix and other capabilities. What you can see on this slide is how we've grown over the years with more than 1,600 companies that we're serving globally and across 60 countries. I think we're at 1,000 employees now, and of course one of our claim to fame is related to novel drugs, in which we are supporting more than 90% in one way or another with technology and or consulting over the past 6 years. Today, we're going to talk about bioequivalence and PBPK, PBPK being physiologically based pharmacokinetic modeling. And so per FDA, bioequivalence are studies to establish bioequivalence between two products are important for certain formulations or manufacturing changes occurring during the drug development and post-approval stages. In BE studies, the exposure profile of a test product is compared to that of a reference drug product. And of course, this is the anchoring scientific point within the generic drug rules, which is you do not need to do clinical studies if there is bioequivalence demonstrated between the two products. Of course, it's easier said than done, and that's where we're talking about where modeling and simulation can play a role. So with that, I'm going to just turn it over to Nikunj for a minute to talk a little bit about our objectives today. Prior to turning it over, I want to talk a little bit and just bring up some terminology for you related to model-informed drug development and model-integrated evidence, which is similar to model-informed drug development, but for generic drugs. So with that, Nikunj, would you share a little bit of this survey that was recently published?
Right. Thank you very much, Ellen, for the nice background. And as Ellen mentioned, the overall purpose today is to go over how we can leverage the model-based simulations to support biopharmaceutics questions or supporting virtual bioequivalence studies. But there are a number of terminologies like MIDD, which is model-informed drug development. There is MIE, which is model-integrated evidence. And I think when we present a number of case studies today, we will bring all of these points and how you can actually leverage those, either modeling on its own or modeling in conjunction with in vitro data. So the purpose of today is how we can leverage PBPK modeling or model-informed drug development to answer and address some of the biopharmaceutics related questions. As Ellen mentioned, this is a very nice survey which was recently done by the Center of Research on Complex Generics. As you can see here in the third column, analytical techniques. So actually in all the three, first, second, and third priority is actually modeling, starting with the PBPK modeling for locally acting drugs. So the locally acting drugs are the drugs when given orally, they would work in the gastrointestinal tract. When they are given dermally, they work on the skin, etc. The second priority is about oral absorption modeling and understanding the orally administered drug for systemic application, for example, and a typical bioequivalence role of modeling in bioequivalence assessment. On the last column, you can also see that the modeling is also one of the priority in terms of education. So people need more education to see how they can leverage this technology, because this is still an evolving field, pretty much new, and a lot of people want to understand how they can use it to solve the problem they face day to day. And one of the purpose of our talk today is to help you understand in what kind of situations we have been successfully able to apply these techniques, and how modeling can be applied in general about the PBPK. Next slide, please. Next slide, yeah. So with that, I would pass it back to Alan to take us through some of the basics or some of our understanding on regulatory perspective of modeling and simulation. So Alan.
Thank you, Nikunj. So as I mentioned a few moments ago, there are some definitions that many of you are familiar with, although there's a little bit of delineation between novel drugs and generic drugs. So in the last few years, there's been a lot of emphasis on bringing these technologies that have been developed and used quite universally in novel drug development over to generic drugs. So the terms are a little bit different, as I mentioned. For new drugs, it's MIDD, sometimes MID3, where the third D is discovery, model-informed drug development and discovery. Whereas for a generic drug, it's more around model integrated evidence. And you can see some slides here that were used by FDA in describing the differences. Now, as we go to model-informed drug development, this is FDA's description, which I'm not going to read the entire thing here, but what it is basically informing is that it is being used in more and more drug applications, and it's advancing its use is part of FDA's program under PDUFA, the Prescription Drug User Fee Amendments of 2017, which will of course be reauthorized next year in 2022, and then followed up with on the similar one for generic. So it basically, there are many valuable benefits here. But these highlighted again by the agency are: one, leveraging a thorough understanding of the drug, the disease, and how the drug affects the human body. These are benefits of using MIDD. The second is to be able to quantify information via the mathematical models, which are based on a range of data sources from in vitro, preclinical, and clinical studies. And then third is how to apply that knowledge. I would say that this is where, really the art form comes in to address issues pertaining to drug development or clinical use. And, there's a long laundry list of uses, which we'll talk about in a minute, but related to identifying what the optimal dose might be, how to determine what the first in human should be, how to provide recommendations for different types of populations, for different types of doses, the effect of pharmacokinetics and pharmacodynamics. And as I said earlier, in addition to PDUFA and GDUFA, the cascade of model-informed drug development has become quite global. Obviously, each agency and each group of agencies manages things a bit differently. FDA has a very sizable group of modelers on staff that do direct modeling of programs where information is sent to them by the sponsor. EMA does more of a review approach. PMDA, it's a mix. The British agency and others have followed FDA in adapting these concepts and encouraging it in use. So as we move down and move closer to our subject today of virtual bioequivalence, you can see FDA recommending a number of approaches and focusing here on PBPK, which is where our case studies are going to be brought to light today. So with that, we look at what are some of the applications. So, first you have new investigational agents, and some of the areas I mentioned earlier. These can begin with formulation, with preclinical to clinical translation, animal to human, well known for drug-drug interactions, and a range of areas for special populations, etc. But in generics, what we see here is that what we're doing is extrapolating a bioequivalence into these different populations, and it's an alternative approach for demonstrating bioequivalence, and increasingly it will be used for developing this safe space for critical attributes of drug products. So, I'm highlighting some of those areas here, but it's really a much broader range of applications. To that end, I'm sharing here a slide that we have developed at Certara. These are some of the compounds that we've worked on over the past several years, where a PBPK, Simcyp PBPK, has been used in lieu of a clinical study. So they've informed the drug label in one or more cases and delivered prescribing evidence to the physician based solely on modeling and simulation using PBPK in lieu of the clinical study. So generally, you do have to perform a clinical study. Let's say it's a DDI clinical study, and then you can develop all other questions and answers that are answered via PBPK, and these are some of those case studies shown here. All of them are available publicly in the literature, and you can see exactly, if you look up in the OCP report, how this PBPK was used in lieu of the study. And then as I pass it back to Nikunj in a minute, we're talking about another case study, this one being for a generic drug, a topical drug. This is really the first example of an ANDA for a generic. In this case, it was a Voltaren topical gel 1%, where the VBE assessment was used, modeling and simulation, and was given the green light without having to do a clinical study. So FDA is quite proud of that, as are we, that collaborated on this, and we see many others coming down the pike. So with that, I'm going to turn it back to Nikunj, who's going to, at a high level, describe PBPK. And from there, we can move into some case studies. So thank you, Nikunj.
Yeah, thank you, Ellen. So the PBPK has become so mainstream term. I think lots of people in the area are familiar with what PBPK is, but just for the benefit of people who are new, I'm going to briefly describe what is PBPK and what is the advantage. So the PBPK, as the name suggests, it's a physiologically based pharmacokinetic modeling. So we describe the reality of pharmacokinetics that we understand into the mathematical equations parameterized by the database of physiology, so that we can simulate what would be the likely rate and extent of absorption of a given formulation when it is given to healthy volunteers or a particular population of interest, as Ellen mentioned. So for example, when we take a tablet, it goes in the stomach, it disintegrates into particles, and the particles dissolve. While they are dissolving, they also empty from the stomach at the gastric emptying time. Then it goes to small intestine, where there will be effect of bile salts, the pH would change, and how that would affect the overall dissolution of the drug. And then the drug gets permeated into gut wall, then goes through liver, where it can get metabolized and then reach the systemic circulation. So all of this is modeled as a system of coupled differential equations. So there are number of rate processes that are happening at the same time or in sequence, and that is based on the knowledge that we have generated about human body. And of course, we parameterize it in terms of not only the average individual, but also the population variability in each and every process, and how that would affect the rate and extent of overall absorption from the drug and formulation. And that is the advantage of PBPK, that we can not only simulate an average individual, but also the population variability, which is very important from bioequivalence point of view, because we are not only talking about the average, but we are also talking about confidence interval. And that's where I think PBPK can give us a more realistic estimation of the likely comparison between the two formulation with the known physiology and formulation interaction. On top of that, as you can see here, this is nicely described by Ping Zhao. It's a relatively older publication, but it's a nice summary of the PBPK. Once the model is there, we can also study some of the intrinsic and extrinsic factors. So the intrinsic means age, for example. So once the PBPK model is developed, we can change the physiology from adult to pediatric, for example, and understand how that formulation or drug is going to behave in various pediatric age groups, as long as we can quantify and understand the physiological differences between the pediatric population of interest and the adults. Similarly, we can study some of the extrinsic parameters like the effect of diet or food. So there are certain drugs which are affected significantly by food, their pharmacokinetics is affected, and sometimes the recommendation is to take drug with food or without food. So with the PBPK, actually we can simulate the fed state or fasted state simulation, or we have recently developed a model which we call food staggering model, where you can actually understand the food and drug administration staggering. So you can say what would happen if the drug is taken 1 hour before the drug, or say 2 hours after the drug, or what happens if you take a glass of water 2 hours after the drug or 3 hours after the drug, especially for poorly soluble drugs. So these models are always advancing to the best of our understanding of the human physiology, and I think we are almost there. We can reasonably capture the likely outcome in vivo based on reliable in vitro data. However, we don't still understand everything about human body, and that's why the paradigm is called predict, learn, and confirm. So the model verification is a very critical parameter that we need to build enough confidence in the model that it is able to answer the question at hand. Next slide, please. And yeah, that's why this is now a flow chart that is copied from a recently published guidance from US FDA on use of PBPK for addressing biopharmaceutics questions. So now you can see there is a regulatory guidance which suggests the maturity of the field. But as you can see here, there are some critical steps of model development and verification. So we need to identify the objectives first. Then we need to make sure that the model options or the model we are choosing is fit for the purpose, that the model is able to answer the question at hand. And then we need to look at the structure of the model, that the model is scientifically sound. We need to look at the parameters, that the sources of parameters are reliable. We need to declare the assumptions of the model and also probably demonstrate the impact of those assumptions on the overall outcome of the model, because if the assumption you are making has a quite critical impact on the outcome of the model, then we need to probably either rectify that assumption or we need to demonstrate the impact of that assumption on the outcome, and that is quite critical. And that all forms into the part of model validation, that we need to demonstrate that the model is sufficiently reliable in predicting what we are actually trying to predict. And obviously there, each and every question and in every drug and formulation requires a different modeling strategy, but overall a good model is scientifically justifiable, mechanistically plausible, and it is sufficiently verified. If you go through the guidance, there is quite a bit of detail provided about what should be the criteria of model acceptance and what can be considered as a verified model. But as I said, it depends on the case by case basis, drug, and the amount of information and the type of question we need to answer. And that's why the last one is the model has to be fit for the intended purpose, and that is the application of the model. Next slide, please.
There you go.
Yeah, okay, thank you, Alan. And sorry, I have to ask you because,
No, it's okay. It's just that it wasn't moving.
Okay, right. And so I think because we are talking mainly about the oral absorption and the oral formulation today, I just want to briefly describe the mechanistic model we got to capture the oral drug absorption. In short, we call it ADAM. So ADAM is Advanced Dissolution, Absorption, and Metabolism Model. So it describes the GI tract into the realistic situation. So when the drug is given, it goes in the stomach. From the stomach, it can empty into the small intestine, goes through various parts of small intestine up to colon, and finally in the feces. But while the formulation is going through the GI tract, it can get dissolved. Then the dissolved drug can get into the gut wall depending on the permeability. It can get metabolized. Whatever is remaining can get into the portal vein and then liver, and then in the general systemic circulation, where we generally measure the pharmacokinetics for a systemically acting drug. And as you can see here, the dissolution, precipitation, the absorption, the metabolism, all of this actually going on at the same time in all the segments, depending on where the majority of the drug is. And that makes the model really mathematically complex. Plus, on top of that, the model is also supported with the variability, and this is quite critical. It requires lots of effort to understand this and derive these parameters from scientific literature. and then make them into usable parameters that can support the model. For example, the radius and length of GI tract, how they correlate with the age and gender or the body surface area of the individual, the pH of the different regions, and if there is any interaction between the regions of the pH tract, how this pH is affected by the prandial state, like the fasted or fed, the bile salt concentration, the blood flows. When we eat food, the blood flow changes, the splanchnic blood flow. So all of these factors are actually accounted for in the model, so we can simulate a realistic scenario as closely as possible when we simulate a model for a given drug and formulation. Next slide, please. Now the critical question is whether how we can parameterize this model. So we know this is the equation on the top is, I know a lot of people don't like equations, but believe me, this is only one equation in the presentation. So this is a nice adaptation of the original Noisy-Whitney equation, which is a Wang and Flanagan correction for a spherical particle. But the particle dissolution rate from a given particle is affected by the effective surface area, which is represented here by AT, which is the particle size at a given time. And also the rate of dissolution is affected by the concentration gradient between the solubility at the surface of a dissolving particle versus the bulk concentration. Now, we can actually use this equation as it is for in vivo simulation, as long as we know the particle size, diffusivity of the molecule, and probably the unstretched boundary layer thickness. And a lot of people actually do it. But if we have some in vitro dissolution data for a given formulation, we could actually use that data and model it in a platform which we call SIVA. So it's an in vitro, SimSip in vitro analysis toolkit. Now, by modeling this in vitro data, we are doing three things. First of all, when we do in vivo simulation, our point of reference or point of verification is generally plasma concentration profile. And we know that the plasma concentration profile is an effect of not only the dissolution from the formulation, but absorption, transit, permeation, metabolism, so on and so forth. So the effect that we are measuring after giving a formulation is much downstream, while the in vitro dissolution is more reflective of formulation. However, we know that the in vitro conditions are not always like in vivo, because in vivo is more dynamic, more complex, and more variable. So we don't want to use this in vitro dissolution assuming that that is going to represent the whole of the GI tract. But on the other hand, we also want to make sure that the model inputs are reliable, the assumptions we are making are correct, and the model is sufficient to describe the effect of formulation and dissolution. If not, then we can go back and measure this parameter again or quantify them. Next slide, please. So with that background, I'm now going to pass back to Alan to describe the background on the case study 1, and then we can go into more detail about what we did. So Alan, over to you.
Okie doke, thanks Nikunj. So we're going to be providing 4 case studies. The first case study is how we use the PBPK model to support and justify a change in manufacturing site post-approval, which is something that happens fairly frequently. I would say FDA provides some clear guidance. There's a lot of information available out here. I've just clipped a couple of things just to give you context. We see here that the types of changes that need to be reported, numbers 2 and 3 are what we're talking about here, which is either the site or the process. The definition of a manufacturing site is perhaps broader than you might think right off the top, because it also includes test sites, packaging sites, etc. And now when we think of the types of changes within a manufacturing site, 1 is a move to a different site for a non-sterile product. 2 would be a move to a different area within the same site. And then 3 would be a site change for sterile products. So to understand a little bit more deeply as to how often this happens, this is a MHRA, the British regulatory agency, a case study of a product that had 54 manufacturing changes over the 7-year period from 2004 to 2011. Now, of those 54 manufacturing changes that were filed with the agency, 30 of them are considered variations that could either require a clinical study or a rationale as to why there need not be a clinical study or analysis done because it's not going to affect the product performance. So while all of them are certainly not ones that PBPK is the right answer for, many of them could be, and that's a significant amount of time, potential time saved, potential money saved, for those changes. I mean, when you think about the case study that we're going to give now is around a manufacturing site change, which a facility was closing, the product needed to be moved to another facility. The sponsor believed the facilities were identical. The agency basically questioned that and asked for a bioequivalence study to demonstrate that that the product would be exactly the same from one to the other. At the time, FDA suggested, well, in lieu of a clinical study, this might be something that PBPK can be used for, and that is how we proceeded from there. So with this just basic picture in the background, if you could describe this case study in a little bit more detail, we believe it's something that happens quite often.
Right. Thank you very much, Alan. Yes, I think the manufacturing changes or after the approval or even during the regular drug development, there is a scale up or there is a slight change in the formulation. And I think all of this typically may require a bridging study or a full-fledged bioequivalence study. And if we already have a validated or verified PBPK model, I think that can be leveraged to answer some of these questions in a scientifically sound way. So before I go on to the case study, I wanted to mention that for this particular case study, we needed a key element of the model, which was we call particle population balance, which we developed on back of the research funding from FDA. So US FDA also encourages and I think allows the further improvement and enhancements of this technology so that they can address some of the complex formulation related questions. So the particle population balance, in short PPB, actually handles the formulation in a more realistic way where it actually simulates a population of drug particles which are given in a solid dosage form rather than older way where either all the particles are assumed of the same shape or size and then all the particles are assumed as probably a smaller range of particle sizes. So here it's more realistic where we account for each and every particles more or less independently or individually and then combine them together to understand the overall dissolution from the formulation. For this case study, we also had to use the food staggering model because the drug had a quite a significant impact of the effect of when the meal was taken, and I think there was a significant inter-individual variability. So we had to also leverage some of the advanced technology that we have developed over the years into the SimSip simulator. So the overall question here was that when the manufacturing site was changed, nothing else was changed. The manufacturing, the API and everything was same, but the in vitro dissolution was significantly different from the historical dissolution data. Now the question was whether that difference in vitro would translate to in vivo or not. So there were two ways to solve this. One, to do a clinical study and demonstrate that the products were still equivalent, even though there was a small difference observed in the in vitro method, or provide a scientific rationale by methods like PBPK to demonstrate that in vitro differences doesn't translate to the similar effect in vivo. And that is, I think, where the situation many of the times where these in vitro dissolution methods are typically developed for quality control. So the purpose is to make them discriminatory, even though they may not be clinically relevant. But then we have to work with the dissolution data, the formulation characterization like particle size, disintegration time, the composition of the formulation to mechanistically and quantitatively translate what we see in vitro to in vivo and then combine it with the population variability to see overall how much difference this minute difference in the in vitro dissolution is compared to the overall population variability of a given drug and formulation. And again, the model validation or verification is quite critical. We had to demonstrate that the model was able to successfully translate the in vitro dissolution of the drug on the historical formulations they had, where the clinical bioequivalence data was available. So we had to build confidence in the model that the model was able to successfully translate these in vitro differences to in vivo, and then apply the model to use this formulation from the new manufacturing site and see how it actually compares with the other formulation. And on top of that, we had to do some sensitivity analysis and worst case scenario simulations to understand the safe space, or basically the edge of failure, or how reliable or to what extent the model predictions could go even when we look at the worst situations of the manufacturing controls of the product. And in this situation, I think the model was positively received, and there was no need to do a dedicated clinical study to support the manufacturing site change. Case
Yep.
study 2, over to you, Alan.
Yeah, well, I was going to just add, a couple of points, just sort of, speak to that a little bit. I mean, it sounds like, first off, there were some nuances to what we had in the model that had been developed over the past few years, really under some of the partnerships and grants that we have with FDA, which is where the PPB particle population balance came in. Because this was a more complex generic, I guess I would say, and that it was demonstrated and FDA was aware of it, which I think definitely helped the situation. The addition being the food staggering, which is of course something that's really critically important to many of these products, and how they're how we can demonstrate success from one to the other as we make these types of manufacturing changes. We don't know exactly how much money was saved or time was saved, but we can easily guess that, several $100,000 and several months. And in this case, the several months is probably most important in terms of if the product had to be either, taken offline or gone someplace else, would have definitely impacted patient's need to get the medicine. So with that, we're going to move into the second case study, which is a corollary, if you will, on the first case study. So, having found success here, that same company recognized that they had a very similar situation, and rather than follow exactly what happened the prior time, where they went and they spoke with the agency and ultimately it was decided that PBPK could be a path to follow, they just preemptively used PBPK, and we got a head start on that in advance of them having to move to the new facility and use the words that Nikunj used before, the learn and confirm from the prior experience to bring success to this one and leverage it. This is also another case that demonstrates from a regulatory perspective how the regulatory teams, they might miss an opportunity to devise a more creative post-approval pathway that can be managed when there is pressure to submit immediately. So, for those regulatory folks that may be listening here, these modeling and simulation approaches can really provide a different regulatory approach and should be considered. So with that, I'm going to turn it back over to you, Nikunj.
Right. Thank you very much, Alan. Yes. So I think if the regulatory strategy anticipates that there is likely use of modeling, I think it is generally better to employ or at least include it into the process as early as possible. So some of the unnecessary experiments can be avoided, or the experiments can be designed that they can be used in a suitable way to either inform the model, parameterize the model, or verify the model. So the earlier it can be adapted into the drug development and formulation development, I think the better it is. So in this case, I think it was a preemptive use, and I think it made the whole process more efficient, more integrated, and also there were less back and forth overall. So that was quite good. However, even though the overall application is quite similar, but the drug and the knowledge about the drug, the knowledge about the formulation, and the clinical data was significantly different than the first case study. And because of that, I think the modeling strategy has to be very specific for each and every drug, each and every formulation, depending on how well we understand the formulation, what kind of data is available to inform the model, and what kind of data is available to verify the model is quite critical to devise a modeling strategy that is suitable for the drug and formulation. So even though the area of application is quite comparable, this was a completely different modeling strategy that we had to apply because the drug had a very complex pharmacokinetic profile where some of the metabolism was involved in the gut wall. So there was some gut wall metabolism involved, and there was a element of enterohepatic recirculation. So again, overall application was comparable, but depending on the drug, depending on the formulation, the overall modeling strategy has to be devised in conjunction and in light of all the information that we know about the drug. Next case study, please.
Okay, so let's switch it up a little bit and talk a little bit about a formulation change for pediatric patients where the tablet would not be appropriate for the peds, for the children. So we moved to a suspension. Now, we all know that children are not small adults, and not all children are the same. And this is really important as we look at developing products, medicinal products for children. Kids may need different doses of medicine. I mean, just today, the data, as we record this, the data about the vaccine for 5 to 12-year-olds came out, and it was a lower dose being recommended by the sponsor. In this case that we're talking about here, it was a different formulation. There's also different types of devices that can be more appropriate for children. And then there's also different therapies at each stage of growth. Things like growth, maturation, and environmental factors, along with factors around the disease and genetics, directly affect drug performance in children. And that is especially true in small children less than 2 years of age, where their ontogeny is different. PBPK is increasingly applied in pediatric drug development. And again, another area where it is encouraged by regulators. Certainly, we need medicines for children, and we need them to be very safe, and we need them to be well explored, but we don't want to do any unnecessary clinical testing. We want to take advantage of whatever information we have in order to develop these drugs and these new formulations and optimize them for these different age groups. So in this next case study, we're going to explore that, and we're going to explore how PBPK was used to change the formulation and gain regulatory approval for a suspension formulation of a drug and expand its usage to children. So there is a lovely picture of a child taking a dose, looking only semi-miserable. Nikunj, to you.
Right. Thank you very much, Ellen. Yes, so I think the suspension has several advantages, especially when it comes to pediatric administration. First of all, you can easily titrate the dose compared to a tablet, where you can either make it half or maybe a little less than half, but I think you can have more quantitative titration of the dose per age group or body weight, etc. The second one is that there is no swallowing involved, so the suspensions are typically easier to administer. But the question is that when you make this change in the formulation, the question is that how different it is going to be from the existing formulation, and I think that's a very critical question to answer. So here again, I think this was another role of modeling, which I think Ellen described earlier, is called model integrated evidence. So here we had to use model as one weight into evidence, along with a variety of in vitro assessment, the prior knowledge of the formulation, the prior knowledge of the other formulations of the drug. So it was actually in conjunction with the physicochemical properties of the drug, the behavior of the drug, and overall understanding, the clinical understanding of the drug. And with the modeling, it added the additional evidence that this change in the formulation didn't have a meaningful impact on the performance of the drug or the pharmacokinetic profile of the drug when it was changed from tablet to suspension, because the tablet was again quite rapidly disintegrating. The primary particles used for manufacturing the tablet and the suspension were comparable. We also did some safe space assessment using the model, where we actually looked at the critical formulation attributes, like for tablet, the disintegration time, the particle size distribution, and some of the manufacturing variables that were captured empirically via dissolution. Plus, on the suspension, we had to see the effect of viscosity, and I think in the simulator we got a model to account for an viscosity effect, that how much it would probably, if there is a delay, how much it is going to be, especially when the drug is being liberated from the formulation in the stomach. Also the effect of the pH of the formulation, because the suspension had to have a certain pH, and then that would affect the overall fraction of the drug remains as solid in the formulation as well as dissolved, and when it hits the stomach, what would be the likely differences, and probably sensitivity analysis around those parameters. So overall, model provided additional evidence and also exploration of the safe space of the formulation to basically support the biopharmaceutics risk assessment of this particular formulation change. And along in conjunction with suitable other evidence and in vitro data, the model was able to support this formulation change without a dedicated study. Next case study, please.
Okay, so this final case study for today is controlling a polymer change for extended release formulations. So extended release drugs offer several potential advantages over other medicines. For example, it reduces the frequency of the dose by modifying the rate of drug absorption. Thus, these types of drugs, extended release drugs, enable higher doses to be given less frequently with less fluctuation in blood concentration. And they're used oftentimes in areas where there is challenge with patient compliance and coherence, ability to stick with the dosing regimen with the extended release, that can be overcome. And so you see more and more products looking to move into this area. This is also often related to, aging. So as we just talked about children and their abilities to swallow, that would be one area where extended release could be of value, but even more so would be in the geriatric patient. And as we all know, geriatric patients often take multiple medicines, and it's a major factor driving the industry. So the lack of adherence of this medication regimen for our geriatrics is very, very common, and this is an area where extended release is definitely growing, as well as CNS drugs. So with that, I ask Nikunj that you share this fourth case study with everyone. Thank you.
Right, thank you, Ellen. So I think this is a case where the modeling can actually impact go, no-go decisions for in-house drug development or formulation development, because even before regulatory, when the formulation is being optimized or developed, still majority of the times people use trial and error. So they try and develop various variants of the formulation. There is now more and more use of quality by design and some of the design of experiments elements, but still most of the time I think it is trial and error basis. So you develop a most possible formulation, go to clinic, test it out in a pilot PK, then probably come back and redevelop it. And I think sometimes it can be quite resource intensive. So here we are giving one example where, I think this was a formulation where the mechanism of release was quite complex. So it was a mixture of erosion and diffusion, and there were at least 3 ingredients which could overall impact the relative rate of erosion versus diffusion and obviously the swelling. So if you look at the trial design of experiment, or even looking from in vitro to in vivo, is quite complex and quite challenging. So here with the PBPK model, first of all, based on the prior information about the drug and the formulation and the clinical data, we were able to establish a biorelevant dissolution method that can successfully translate probably quantitatively or semi-quantitatively in vitro evaluations to in vivo. So the model was able to identify the biorelevant dissolution testing method so that it can be employed for this evaluation of the various variants or of the new formulation. And with that, I think the model was able to identify what was the more likely outcome of the formulations that were developed, and then it basically helped expedite the decision making and probably number of trials and error that they had to do. And when the formulation was tested in the clinic, the predictions from the model based on this biorelevant method were very comparable to the outcome that they observed in the clinical study. So with that, I would pass back to Alan to summarize our talk and provide some useful resources. Thank you.
Thank you. So we've covered a lot in a short amount of time. We've given 4 case studies just to sort of make the point that there's a wide range of opportunities to use modeling and simulation, and specifically PBPK, in demonstrating virtual bioequivalence and in bringing generic products to market, especially complex generics, which is an area that we've been focusing on alongside the regulators in the past several years. We like to say that these methods, whether it's PBPK or PBBM, many acronyms in this business, have evolved from a research nicety to a powerful research approach for both regulatory perspective and bringing, as I said earlier, new paradigms to the regulatory process, and for R&D in biopharmaceutics risk assessment. What we have, I believe, demonstrated today is that PBPK, scientifically sound PBPK models can expedite internal decision making. They can enhance, as in the last example, a go, no go. They can help you choose the right path to take. They can help you with patient selection, etc. We can be using them for changing formulation development from the old trial and error-based evaluations. It never ceases to amaze me, even today, how doses are selected in a more random way when there is quantitative methods such as modeling and simulation, such as PBPK, such as QSP, etc., to help select the right dose for the right patients, and then shifting development to a much more scientific-driven paradigm. We see this going on every single day, and we're very encouraged and excited about that. The third point here is there's a belief with PBPK that I want to quell right now, which is that it's for DDI only, drug-drug interactions. And of course, for drug-drug interactions and special populations, PBPK is optimal, and we recommend it often and have many ongoing projects in these areas. But it's also for other purposes. And today we spoke about how PBPK can be used for formulation development, for generic drug approval, for the demonstration of virtual bioequivalence, for alternative regulatory strategies. They can be used early on in the drug development program for both innovative companies and generic companies, not just in late stage DDI. So I cannot emphasize that enough. It's also being used for new modalities, especially biologics, and we're happy to provide more information should that be needed. There is always a need to share more and more. We at Certara publish, as well as our academic partners, we have well over 100 academic partners that use SimSip, and they publish, and this is a way to speed awareness and to share the latest that's happening in this industry. To that, I have provided a list here, which is, of course, these are all links. If you can link directly from this deck when you download it, of some of the examples that we talked about today. Of course, we have many others that we're that were published and are published on an ongoing basis by ourselves, of course, but most importantly by the regulators and by our academic partners. So on behalf of Sutara and from Nikunj and myself, thank you very much for attending today's session. We hope that you now have the confidence to take that next step and evaluate the use of PBPK for determining virtual bioequivalence or any of the other areas that we touched on today, and look forward to working with you in the future. Thank you and take care. Bye-bye.
Yeah, thank you. Bye.